So good afternoon, everyone. Welcome to the sixth mentorship lecture of the Eurocolangionet Action ENSCAN ICRN on behalf of Chiara Gabi and myself. I'm Rocío Macías, and together with Chiara, we are organizing these mentorship lectures to help the career development of young investigators in our network related to cholangiocarcinoma. We want to thank our speaker, Professor Moser, for accepting our invitation to participate in this activity. As you know, these lectures have the endorsement of the European Association for the Study of the Liver, EASL, and the United European Gastroenterology, UEG, and also have the support of our partners, the AMMF, the UK Cholangiocarcinoma Charity, and the Cholangiocarcinoma Foundation, the US charity. And we also want to thank the University La Sapienza of Rome for hosting the lecture. We have here with us Professor Vincenzo Cardinale, the Action Chair of Eurocolangionet. Hi, Vincenzo. It's my pleasure to pass the word to you.
Thank you, dear Rocío. It's a great pleasure for me to host as a chair of Eurocolangionet and also as a member of Sapienza University of Rome in our platform, this very precious webinar. I have to thank, first of all, the organizer, Rocío Macías and Chiara Gabi, and of course, our very eminent invited speaker, Professor Vincent Moser, and also last but not least, our young investigator within this network that will make a question. So it's a great pleasure and honor. This initiative, the mentorship program, is the one of most important initiative in Eurocolangionet, and I really hope for the future of this initiative in next grant. Thank you.
Thank you, Vincenzo. So now let me introduce you our two moderators who are members of the Young Investigator Group of Eurocolangionet. Dr. Paula Olaizola is a postdoc in Bolter Lab in Edinburgh, and Ravi Jagetia is a PhD student in the Chokshi Group in London. And thank you both for agreeing to participate as moderators today. I give you the floor. Thank you.
Thank you. Hello, good afternoon, good morning to everyone. First of all, I'd like to thank Chiara, Rocío, and Vincenzo for the kind invitation. It is a real pleasure to be here and to introduce such an excellent speaker today. So Vincent Moser is a physician scientist. He holds since 2019 the Canada Excellent Researcher in Genomic Medicine at the McGill University in Montreal. His 40-year career has been driven by his passion for pharmacology and genetics, and more specifically, the question which has served as his compass has been, how can human genetics support the discovery and development of new therapeutics? During his mentorship lecture, he will share his single question-driven, unusual, yet very enjoyable journey in biomedical research at the clinic, at the interface between academia and industry.
We'd just like to let the viewers know and the attendees that the chat and the Q&A section is open for questions. So as the session goes on, if you do have any questions, please feel free to populate them, and myself and Paula will be reading them out at the end. And that just leaves us to invite Professor Moser to share his screen and crack on with today's great presentation.
Can you see my screen well? Okay, let me first thank you first. I really appreciate to be invited here, and I was looking forward actually to talking to you and maybe sharing some of my experience. I'm probably in the last stages of my career. I've been very lucky in my career. I've spent lots of time in academia and industry, and I'm now also in a very delightful position here at McGill. So just want to share with you my experience. I am particularly appreciative of the invitation because I'm not by no means an expert in liver diseases, even less so for cancer. But I've been in industry and I'm happy to share this experience. What I'm planning to do is first sort of set up the landscape. Where is the field? What is the field I'm working on? And then I will share with you my sort of trajectory in this space, and then some thoughts which sort of have been guiding me along this journey. And of course, I look forward to having the Q&A session. This picture is from a drug and dates from more than 100 years. You can see here the name of the drug, asthma cigarettes. You can see the indication, the temporary relief of paroxysm of asthma. You can see the extended invitation going from asthma to hay fever, foul breath, and even cancer. You can see the contraindication, not recommended for children under 6. And you can see the manufacturer, Dr. Batty. Luckily enough, things have changed, and it's much more complicated now to bring a new drug to the market. The different steps which are now necessary to go along are illustrated in what we call a chevron diagram. It starts with the far left side with a disease. You need to pick a disease you want to develop a drug for. Cholangiocarcinoma, for instance, or coronary artery disease. You then need to identify a target. That means a protein, a receptor, a nuclear factor, something that you want to modulate so that you hope you will change the trajectory of the disease. You need to bring data to substantiate this target, and so what we call target validation. And then once you have identified your target, validated it, you need to go into chemistry, high throughput screens, find hits, which you will, thanks to medicinal chemistry, improve to get a candidate. And once you have selected the candidate, which has already met a whole series of filters, you go into human, FTIM, first time into man, and firstly in volunteers to demonstrate what we call tolerability and safety, and then into patients to demonstrate proof of concept, POC. And once you have demonstrated that your drug works in a few patients, you go into larger trials called phase 2 and phase 3, and then you get regulatory approval and can enter the market and go into life cycle management. All that is long, risky, and costly. And I'm just taking here this comparison to give you an idea of what we mean here. I compare here the costs of discovery and development for the latest Airbus airplane with the usual drug. Costs of development for the airplane were $10 billion US. We're talking about $1 to $5 billion US for a new drug to come to the market. Duration is very similar, actually 10, 12 years. What is different, though, is probability of success. Normally, when a company launches a new plane program, they have high expectations and, honestly, a high chance of flying for this new plane. Doesn't mean a high chance of making money, but at least a high chance of flying. This is not the case for drugs. It's estimated that 1 out of 20 drugs, which is tested into humans, healthy volunteers to start, only 1 in 20 will eventually reach the market. And that's why people are quoting here this sentence, drug discovery isn't rocket science, it's harder. And that's really correct. And people who have been lucky enough to be in industry all make this experience. So that's the landscape I have been working in for the past sort of 30, 35 years. And if you don't mind, I will now sort of rewind and describe a little bit my trajectory. Not saying that my trajectory is exemplary, by no means. I'm just have been very, very lucky to go through this trajectory, and I'm just happy to share some thoughts. I'm an MD by training. I did my medical school in Lausanne in Switzerland, so not far from Italy. I subsequently acquired a full training in internal medicine. So I'm board certified in internal medicine. I've been very lucky to do that because it gave me an exposure to a broad range of medical specialties. I had acquired also that way a broad culture in medicine. But really what intrigued me from very early on is drugs. And really an interest in pharmacology, which I started to nourish first in my first research job, which was at the University of Lausanne Hospital Center with a guy called Hans Brunner. Hans Brunner was the first person to test ACE inhibitors, so captopril at that time, into humans when he was in New York. So he was very famous already for all his studies, proof of concept and healthy volunteers that he had been running on this new class of drugs. And that really excited me and prompted me to ask if I could spend a couple of years in his lab. I did actually a trial there, or run a trial with an ACE inhibitor from Roche at that time in healthy volunteers. That was lots of fun, lots of learnings as well. I subsequently decided to go further into pharmacology, but this time moving from human pharmacology to experimental pharmacology in the rat. And Hans recommended to me a lab in Australia. And that's where I did my first postdoc. It was 2 years at the University of Melbourne under the leadership of Colin Johnston. Colin was very famous for his work in experimental pharmacology on ACE inhibitors. I spent 2 years there, got to learn as well the other guy here, Stephen Harrop. I need to tell you this story. Stephen was working next to me and had just come back from Oxford, and he was studying the genetics of baldness. We're talking about the late 80s. I found it very weird, very strange. But Steve told me, oh, it's not so weird because baldness is a risk factor for cardiovascular diseases and has some connections so with our disease of interest. So I remember that very well. And actually, you will see that this discussion with Stephen actually prompted some interesting moves in my own career. I came back to Switzerland with our first child, actually, from Melbourne, and completed my internal medicine training. The training in Melbourne was fantastic. I learned a lot, but honestly, I did not learn enough to be an independent investigator, which was really what I wanted to be in the field of pharmacology. And I was actually chief resident in the hospital there in Switzerland. And I was sharing my concern with a representative from a company called Park Davis at that time. And this woman told me, I think you should go into genetics and lipids. We're talking about 1991. And I didn't understand what she was saying, but I was intrigued. And she was telling me, you know, we have some new drugs coming soon into the market. Park Davis was the company which discovered atorvastatin, which is a very effective statin. Company was eventually bought by Pfizer. And she was saying lots of stuff are cooking in genetics and lipids. You may want to go into this field. And I was very intrigued and started to talk to people around me, started to search the literature, and she invited me actually at a conference on genetics and lipids. I remember very well, it was in Münster, Germany. And there, wow, I had this wow moment. So many things were happening. And so I decided to go for a second postdoc and decided to this time go to the best lab in the world. And so I took my pilgrim stick and decided to visit 4 labs. One lab in Dallas, Texas, one lab in San Diego, another one in Seattle, another one in Boston. And I spent 2 days in each of these labs, wondering whether that was the place for me to really learn genetics and lipids. And there was only one lab which really came out of these 4. It was the lab of Mike Brown and Joe Goldstein. These 2 guys had just been awarded the Nobel Prize for their discovery of the LDL receptor. And they agreed to have me as a postdoc there to work on a particular lipoprotein called LP-little-a. My direct supervisor was Helen Hobbs. And sorry for the picture, it's terrible, but I just wanted to actually highlight what is on the top there. Helen has become a giant in medicine. And she agreed to have me in the lab. She wanted me to work 6 days a week. I had already 2 children. We negotiated 5.5 days. So I had to work on Saturday mornings. And that was a crash course on genetics and lipids. Amazing. We had our third child there. So first in Australia, second in Switzerland, third in Texas. And eventually, having acquired a strong background with Helen's supervision, decided to go back to Switzerland to build up my own lab and worked with my buddy there, Gérard. Gérard and I had been buddies since the age of 16. So we were together at high school, and he was an endocrinologist. He's an endocrinologist. We decided to join forces with my background as lipidologist and he's as diabetologist. And we decided to run some large projects together, trying to really use what I've been learning in Dallas, Texas, and build up some new projects to better understand the genetic basis of diseases. And that was at that time that I got in touch with Alan Roses. Alan was at Duke, and I got to know his work when I was in Dallas, Texas, because he had just discovered the association between a lipid gene called ApoE and Alzheimer's. And he had just been hired by GSK to build up some new research program based on the same paradigm. That means, how can we use genetics, like he had been doing, ApoE and Alzheimer's, to better understand the molecular basis of diseases? And Alan was looking at centers around the world to collect samples from patients with a variety of diseases. I went to meet Alan at Duke at that time and said, I can collect for you samples with coronary artery disease, and that's what we did. It was a very big effort, but rewarding. And when we had done this project, Alan came back to me and said, I want you now with us at Glaxo to run the genetics of cardiovascular diseases. Of course, very difficult decision to make, 3 children, a wife, going back to the US, this time to Philadelphia. I said, okay, I'll try. Couple of years, we'll see what I can do. Eventually, I stayed for 10 years at Glaxo. And I was very also puzzled, because mandate I had been given was find new genes for cardiovascular diseases, but we had samples, but not enough samples. And I said to Alan, we need to have more samples. We need to build up a population-based study whereby we recruit thousands of people, very well characterized phenotype, but also examined with the latest genetic technologies. And Alan said, yeah, I think you convinced me. Do it. Here is a check, do it. And so with my buddy here, Gérard, you have seen before, we built up one of the first population-based studies actually in the world. It was 2003. Now these studies are dwarfed by large cohorts like UK Biobank or FinGen, but at that time, that was one of the earliest. And actually that allowed us to do some important discoveries. We were among the first ones to discover new genes associated with LDL cholesterol levels, for instance, when you can see here. Talking to this audience of hepatologists, we were also the first group to report the new gene for NAFLD, for NASH, called PNPLA3. And that was done really using this database that we had created. When we created this database, I also remembered Stephen Harrop's. And I thought, one, when we collect all this data from these 6,000 people, let's collect data on boldness. Let's ask the question, do they have boldness? When did it start? And even actually sort of quantify boldness. And that was unexpected from the investigators in Lausanne I worked with, but they eventually did that. And once we had the genetic data in the computer, we had the boldness data, we identified actually these locus here, this region on chromosome 20, which for the first time had been discovered to be associated with boldness. On the x-axis, on the very far right, you can see the androgen receptor. So that was really cool. But eventually, when you do this type of GWAS analysis, that means genome-wide association studies, whereby you can interrogate, like is the case here, millions of variants or SNPs along the human genome. When you do that, there is also a risk of false positives. You test essentially 1 million hypotheses, and what you have to do is a replication set. And when I was looking for a replication set, people were telling me, oh, call this guy, Brent Richards. He's at McGill, and this guy has a data set which could be useful for your replication. And so I called this guy, didn't know him, called Brent. Brent said, yes, give me the number here of the variant, and I'll get back to you whether it is associated with boldness in my data set. And took him a week, and 3 months later, we had a Nature Genetics paper where I'm the first, Brent is the first author and I'm the last author. So that was really cool. I decided eventually to stay at Glaxo because I really enjoyed that. And I really enjoyed the environment. I moved up the ladder there and became vice president in charge of applied genetics across the entire company, with 2 outstanding leaders, Paul Matthews. Interestingly, Paul left Glaxo, is now in Oxford, and is the head of imaging for the UK Biobank. He is a very prominent researcher in the UK. And Lon left Glaxo eventually to take the role of chief scientific officer and chief strategy officer for a company in California called Biomarine. He's now the chairman of the JAX laboratory in the US. So really enjoyed that, but eventually family reasons and other reasons, I was sort of headhunted back to Lausanne and accepted to take this role, which was actually quite administrative, leading a group of 600 people, which gave me as well the chance to build up some new thing. And I was vice dean for research there as well. And I had built with Gérard this COLOSS study, the population-based study. I had been really working at Glaxo on also using genetics for patient recruitment. And so I convinced the organization there to build one of the first hospital-based biobank, which we call the Lausanne Institutional Biobank, which would be used as well for selecting patients for trials. It was great. We built up this cohort of patients in 3-4 years, recruiting 40,000 patients. So that was quite unique, really. But then sort of a hiccup came up. There was a new dean. He was not interested in this project, and was making my life a little bit complicated. So instead of going against the tide, I accepted actually to apply for this position at McGill. And McGill was looking for somebody who had industry experience and a track record of building up things. And this guy, Brent Richards, whom I had worked with 10 years earlier, came back to me and said, please apply. We want your expertise and build up the same thing you have been building in Lausanne. We want to make it happen here. And we have the funding to do it at McGill. And so in 2019, I decided to move to McGill. I built up this new team with a very good governance. And look who agreed to be part of the international SAB. Helen, who was my mentor 20 years ago, and Len Carden, who was my mentor in my N+1 at GSK. So that was a spectacular team in the SAB, Scientific Advisory Board, that I had. Everything was fine and moving fast, and it was great. But then eventually something happened, which you can recognize here. And here, and something I think funny, the provincial government in Quebec came to me and said, we know you just arrived in Quebec. We know what you have done. Why don't you build up a COVID biobank for Quebec? And I received this mandate, sorry, it's in French, on the 19th of March. So 11 days or less after COVID was declared a pandemic. This guy, the chief scientific officer of the province, gave me money, gave me also the real mandate to build that up. And we built these in a matter of days across the province. We did have very fast track ethics approval. Again, we were looking what was happening in Italy. We were looking what was happening in the UK. We were just 10 days behind or 2 weeks behind. And so we had this sense of urgency. Let's make it happen. And so 1st of April, Brent Richards, again, the same Brent, recruited the first patient for the COVID biobank in Quebec. You can see that within a matter of weeks, we recruited 1,000 patients. And we did have and generate the genetic data, which very rapidly gave us the opportunity to work with international groups around the world, including the Host Genome Initiative, and be part of big consortia with the discovery of new genes explaining part of the inter-individual susceptibility to COVID and its severity and long-term consequences. So it's been really fun, working really hard. But now that I've stepped down from this COVID project to really refocus my attention on something which Brent and I have been working on and for years now, is moving from COVID to diabetes and complications. So we are now building up a large biobank called Bioportal at the Jewish General Hospital here in Quebec, which is part of McGill, and have received now the funding and the ethics to start the recruitment of patients, to really try to better understand at the molecular level what is happening in terms of diabetes and complications, and hopefully find new treatment for these conditions. So that was my trajectory. It's a bit winding, but I've been very lucky and very happy all along these years. If you don't mind, and that was really prompted by a discussion I had with you, Chiara, and the students, I'd like to share with you some thoughts maybe on this trajectory. And these are just pictures that I've taken from the web. This one here. I think I can probably claim that these 40 years were all driven by one question. Yes, that means, how can we contribute to a very difficult endeavor, which is the discovery and development of new drugs? Of course, my contribution has been minimal, and I think probably sobering as well, despite all this work and all this money I've spent with these cohorts. But still, it was driven by this question: where is the North? And I think that there is probably something that can be learned from my humble trajectory, is the importance of having these Norths. What are our values? Why do we fight for? What wakes you up at night and gives you the energy to work the whole day? So I think it's really important to have that. Actually, you can see as well the little motif here: life is a journey, not a destination. And that takes me to the second picture. It's a journey, but it's a long journey. And I like to take this analogy. We're starting to work, so around 23, 24, when we complete our university degree, we had 42 years to run. That's a length of marathon, 42 kilometers. And we'd better be, firstly, very well trained. Second, have some very good equipment. That means shoes. Thirdly, having probably someone to help us as well, prepare up to this marathon. Otherwise, you get hit. And everybody talks about this wall that marathonians are experiencing around kilometers 30, 35. You'd better run over this wall and so be very well prepared, in my opinion. The other thing is I've been taking risks, probably lots of risks and maybe too much risk, but every time you take a new job, every time you go to another continent, every time you're starting a new experience, you're taking some risk. In this particular case, sometimes it was really swim or sink. I've been very lucky to swim every time, but I think that we need to be brave, and especially if we have the ambition to have some impact and use the best of what has been given to us, which is life. So I've taken risks, and I can certainly support what Paolo Cello was saying. That means nothing can substitute experience. You've probably seen from my experience as well that I've been very lucky in building up international networks. These folks I've met in Australia, in Switzerland, in the US, in industry. You can see that now I've got Helen Hobbs, my previous mentor, being chairing actually now also my SAB. So you've seen this story about with Brent Richards. I got in touch with him just for a scientific reason. He eventually is the guy who prompted me to join McGill. So networking is absolutely essential, but it's hard. Networking is just simply wishing happy birthday to people with an emoticon. It's more than that. It's an active work. It takes time. And I think it requires a very proactive and dedicated effort. Something which I've been learning as well in industry, which seems to be intuitive, but it's not that easy, is listen. I think I've been very lucky that I've been listening to people. I was listening to Stephen Harrap when he was telling me about his boldness story. I've been listening to what other people were doing in the company when I was at Glaxo. At Glaxo, I remember this guy telling me, you know why we have 2 ears and 1 mouth? It's just because we need to listen twice as much as we talk, which I think is very true. We're talking about mentorship here, and I think it's a big effort to find the right mentor. You cannot imagine how much I owe now to Helen Hobbs for my career. I joined her lab in 1993. We're still friends working together. We had a paper recently accepted for publication in Nature. She actually proofread it. She edited it. She was with the red pen and destroying everything which had been written. So this is the type of mentor that you want to get. And these are not easy to find. It's an effort to find them. I think probably the right thing I did when I decided to go to the second postdoc is really go and visit 4 labs and take the one which appeared to be the best for my career, my ambition, but also the compass which I had established. I think it's hard and we need to be passionate. I've been lucky because I had this passion which has grown over years for genetics. Genetics is an area which is certainly growing and fulfilling for me because there's all these new technologies and genetics has been great as well in terms of enabling large networks around the world. So it's been good for me, and I've been, again, very lucky to pick this field. I'm taking this here because I think it illustrates that it's not easy. We need to have some luck. We need to pick our battles because we need to juggle with a whole series of things. We need to juggle with our career. We need to juggle with our networks, with our friends, with our parents. But I think there is one thing where I think I did right, I think, is really cherish my family. And I've been very lucky. This is my wife, and she agreed to go with me to Australia, to the US, back to Switzerland, back to the US, and so forth. And I think I've been very lucky as well to have her. And we have these 3 children. So it's been great as well. So that's my little story. I'm done with my little story, and I don't know if you have any questions.
Vincent, well, firstly, thank you so much for sharing your story and your career so far. I think it's amazing to sort of hear your perspective and the way you look at your outlook on your career as well, retrospectively looking back at how things have gone. Because I feel like as early career scientists, we're kind of in the journey and we don't foresee where it's going, and we don't know what's there's not been a lot behind us to look back on. So it's a very sort of intriguing place we're in. It's nice to kind of retrospectively see your journey as it kind of happened. So I think Paolo should be coming online soon as well to kind of ask. So I just want to remind everybody to get the questions in the little Q&A section at the bottom, and then me and Paolo will be moderating those. But to get the ball rolling, I have many questions. I could probably ask them for at least half an hour, but I think there's 2 that I'm really interested in asking you. One about your career, and then maybe one about more specifically the science that you've kind of been doing. So I'll ask you my first question. A lot of people talk about the benefits of having a career in industry, whether that's private healthcare, so on and so forth. But so as somebody who's been in academia and then gone to industry and come back to academia, what are some of the benefits that you can outline to us as junior scientists that are associated with a career in academia, and what kind of led you to, come back from industry where there may have been more perceivable benefits of having a career?
Yes, thank you, Javi. I think it very much comes back to what are we expecting from life, essentially. I was fascinated by drugs. I came up to this point where I realized that there is no drug which has been marketed yet out of university, out of academia. It's not our job. If I wanted to really understand what it is to develop a drug, I need to do the step and go into the dark side of the fence, some people would say. I did not regret a single minute to have spent these 10 years there. I call that my 10-year sabbatical, because being exposed to really this life of these people and how hard it is to bring up a drug to the market was just, for me, illuminating, and meeting so many people. Of course, there are some advantages and disadvantages in industry which we can discuss. Eventually, for me, it was also clear when I left in 2011 that industry is coming up with lots of new drugs now, which need to be tested into humans. And testing into humans is done by people in academia, the people who have access to patients. And so I thought, well, it's a natural step now to go back to academia and build up this resource, which I have mentioned to you, this Lausanne Institutional Biobank, which was specifically designed to develop new drugs, not discover them, test them, develop them into patients. And that's what I'm building up now this resource at McGill. So it's a sort of a very natural trajectory. It was not driven by my appetite for money, because that was not my first value. It was not driven by nor push from being rejected from a work, but it was really driven by my appetite for science and my chance that I was lucky enough to be healthy, to have a family which was supporting that, and to try industry. So I have no regret at all of what I've been doing. Had I stayed in industry, I would have now a different trajectory for sure. I think it's a matter of taking risks, but also weigh very carefully these risks, because every step is a risk. I will describe now a bit industry. I loved industry again. I joined industry as well when I had already I was not a heavyweight by no means, but I was not a lightweight either. That means I had already accumulated some experience, and I could demonstrate that I was able to do stuff, which industry values essentially. And so I could enter at relatively high level, already director, and go into vice president a few years later. That was helpful. Industry is looking for leaders. Industry is looking for people who have some forward-looking capabilities. Industry is looking, honestly, for medics as well, which have a relatively broad view of things. But it's not the panacea. That means during these 10 years, I had to reapply to my job 3 times. So one day they decided they were on the verge of stopping all genetics, because they were saying, do we really get what we hoped to with all these investments? And so they were on the verge of cutting down genetics and me losing my job. They've actually fired the people above me. I was just underneath the cut. So I was lucky to stay there. And industry also had this way of reducing the number of people. So 2 times I had to reapply for my job, musical chairs, which is not very pleasant. So industry is great. There is money. I could never have built these projects like the Lausanne Color study without industry money, but it has got some risks as well. And that's why it's so important to stay healthy and well balanced, and again, make every step to stay healthy. That means exercise. I exercise 3 times a week, and I'm sure you do as well, but it's just hard to find the time to do that when there's so much pressure to succeed. So I'm still trying to do that. I ran my first marathon when I was 57, just because I thought, wow, it's important to stay healthy. So that's a little bit my story, Ravi. I don't know if it answered your question.
Yeah, I think it did. And just for my second question, briefly, before we sort of, Paolo can maybe ask a few as well. A little bit more related to your career and some of the stages you kind of talked to us about from the journey as a drug from initial conception through to sort of, authority sort of FDA approval. With regards to, I just wanted your general opinion on this again. With regards to clinical trials, they tend to really pick a very niche cohort of patients, especially, in the case of primary liver cancer, we know that the clinical trials choose patients that have very good liver function, and this is affecting sort of the outcome of the clinical trial. And then when these drugs are sort of getting the FDA approval and they're now being trialed on a full cohort of patients, ones that may have diminishing liver function, the sort of effects of these drugs that we're saying aren't as potent as what the clinical trial represented. I wanted to ask your opinion with regards to, do you think clinical trials need a more balanced cohort of patients that represent the entire disease? Or do you think that there's other areas where maybe the development of a drug could be sort of adjusted to kind of lead to less failure when it comes to the application of drugs in patients?
I think diversity is really important here. You're mentioning degree of severity of liver dysfunction. I can also immediately respond to that gender as well. That means we know from that the woman is a very small minority of patients enrolled into clinical trials. Do we need to address that? Yes, we do. But I would say that the very early stages of drug development, that means proof of concept, I push for these studies to be sort of very focused, just to have a quick answer. Does the drug work or does it not work? If we have these clear, precise answers, sort of black and white answer, which we never have, but that requires highly selected patients. And if the drug works in these patients, of course, development needs to be broader. But I think it's unethical to expose people, large number of diverse people, to a drug where we don't have some sufficient confidence that it works. So that would be my answer. The second part is that companies are very aware now as well that their drug does not represent necessarily the right patient population. And so these real-world evidence, which they call now, that means accumulating data from real world, is very important, and will also, I think, be more and more used, especially now that we also have better ways of quantifying effects. That means we've got all these digital solutions, wearables and stuff. So I think that industry is moving into this direction. I think it's a great direction that industry is moving into. And I think regulatory agencies also are now looking at this type of real-world evidence more and more closely for their approval.
Thank you.
Hi, thank you, Professor Musa. That was an excellent talk, very inspiring. And I wanted to ask you more about how has your experience in industry helped you in your academic career and vice versa? And also like linking it to a question we have from the audience, like what is the main difficulty you had to face during that transition between both?
Okay, so the first thing is there are some real commonalities, and I want to emphasize that, between what I've been doing in academia and industry. It was all driven by a single question. That means, how can we use our knowledge, our new technologies to sort of support the discovery and development of new drugs? So there is no real separation between the two. Of course, I was paid one day by academia. I needed to publish, I needed to raise money, to get grants and all that stuff, which I've done. In industry, you need to demonstrate how much value you bring to the pipeline, essentially, to portfolio. But eventually, it's not that difficult. It all relies on good science. That means industry relies on good science. They invest so much money. If you have a shaky target, if you have a shaky molecule and you invest into it, you will lose money. So you need to have the best science to support each of these decisions along the pipeline. And best science is acquired, and people need to learn these skills in academia. So that's why I think it's important to have a strong academic background. It's good to be exposed to people. It's good to fail and have the resilience to come back. These skills and sort of mindset are very useful in industry as well. What industry has brought me, which was very helpful when I decided to go back to academia, is first management skills. So industry relies on good managers, leaders and good managers. They train people. And I was well trained, I suppose, to lead a group. All these things seem very intuitive. Yes, it is intuitive, but we need to acquire these tools and accumulate these toolbox so that when we are exposed to leading a large group, like I had 600 people in academia, you sleep well at night. You don't have these headaches that you can be exposed to if you don't have these managerial skills and tools that you have developed. So I love that in industry. I love the network as well. I was very lucky because I would never had been exposed to such a broad network of outstanding geneticists in the world, being in academia and in industry. Had I not been there, have I just continued my work in Lausanne. So that was a blessing. Industry brings as well resilience. That means when you are in this unpleasant situation, when you have to reapply to your position, you need to keep calm and think and say, okay, that can happen. The last thing is there is always a solution, as long as you're resilient and you're happy. And when I was facing some front wind in Lausanne with the latest step, I could just say, okay, again, instead of going against the tide, I can go to another job. And I've done that before, so I've done that again. So that's a little bit what I've been learning. But I've been lucky, and again, I think it's just like diseases, genes, environment. I've been lucky to have good genes. I've been lucky to be in good environments, and there are some stuff that you cannot control.
Thank you.
So just like to remind people, if they'd like to ask questions, please do get them in the Q&A section or the chat, and me and Paula are more than happy to read them out. So Professor Musa, I had another question, since we're having this sort of discussion about your career and the trajectory it's taken. And you put a lot of emphasis on the role that your mentors had in your career and progression, and they're still continuing to have, whether that be in the capacity of a mentor or now even a friend. What are the qualities that should be sought, not in a mentor, because we know it's quite well known what a good mentor should bring, but what are the qualities that a good mentee should look to bring to the table in a relationship between a mentor and mentee? I think you touched on some of them towards the end of your presentation, but it would be nice if you could elaborate on some of those. Like, what should a person seeking a good relationship with a mentor be bringing to that relationship almost?
Yeah, mentors and mentoring is terrific. And that's really something that I would encourage everybody to have at any level of your career. I'm still now talking to people and asking for advice, because it's hard. There are so many things which we don't see, which are in the blind spot. And these people see that. So firstly, a mentee needs to pick the right mentor, somebody who is knowledgeable, who is ideally in a different field. You don't want somebody who is doing the same thing as yours, who views stuff which you don't see yourself, who is generous, because it requires lots of generosity, and who is passionate as well. As a mentee now, beyond the fact that for finding a right mentor, it means getting proactive. Mentoring is not a passive activity. It's a very proactive activity requiring from the mentee to get ready whenever they meet the mentor, to prepare up these meetings with the mentor, with an agenda, to follow each visits and discussion with progress report. So that's, I didn't know that before at all. I didn't even know the word leadership, no mentoring when I was a student. I've been learning that probably the hard way. But I think now young people are much better sort of suited and equipped than we were when I was younger. But these are terrific tools that, and of course, new technologies enable now to have these discussions around the world. So it's not necessary to have a mentor next door. So that would be my response.
And with regards to, as you said, the mentee should make a active effort to seek their mentor. What would be the best way? For example, this is more of almost a personal question. If you have identified someone you'd like as a mentor, how is the best way to approach this scenario in terms of initially connecting with that said person? is it always better to kind of do it in person, or now, like you said, with advances of technologies, could this be a simple conversation via email? And how would you approach this scenario as a junior scientist looking for who you may think is a good mentor?
I would say it depends. If personal meeting is possible, I would certainly favor that. Now, if you identify a mentor which is remote, you probably want to have our first conversation by Zoom or technologies, and then possibly have at a meeting, say in a scientific conference, for instance, somewhere. But we need to build up this personal relationship, and the mentor should know well the mentee should know well the mentor, shall know everything that the mentor is doing. Again, it's a very proactive activity. People frequently, and that's something I tell to my children as well, underestimate how proactive networking needs to be, mentoring needs to be. It's hard work. And I think it comes back to this question I think that Chiara is asking, or somebody in the chat.
Yeah, so I'm happy to read them out to you.
Oh, please,
so
please do, yeah.
Yeah. So in the chat, we have a couple questions. The first is asking for tips on how to balance their personal life, especially when moving internationally, as you did so many times, as you mentioned, back and forth. And then I'm happy to read out the second one after we've gone through that one.
It's hard. I have no secret. I don't know. But I think it's really important that your spouse and family are enthusiastic as much as you are in making this move. People say, and I think they are right, happy life, happy wife. So, sorry, and then you can turn that upside down. But it's really important. It should be a decision which needs to be discussed very extensively and planned as well with your spouse or partner, because every move here entails difficulties, expenses. We are losing money every time we move. I think it's fair to say that. And if there are children, it's hard. That means I took my children off school when we moved to Philadelphia. They were 10, 8, 12, 10, 8. It's hard. They didn't speak a word in English. They had lost everything they had learned in Dallas, Texas. And then when we came back to Switzerland, they were 21, 19, 17. It's not easy, and they hated us. So sometimes our children for forcing them to do these moves. So I would just say lots of discussions and really planning and avoiding surprises. Yeah, but so I have no secret here. I've been lucky. I wish people to be as lucky as I have been. Yeah.
And then the second question in the chat is regarding job openings in industry, which often require previous industrial experience. So how does one become a competitive applicant when they only have academic research experience when applying to jobs in industry?
Yeah, that's a very good one. In my particular case, I was quite lucky again, because Glaxo was looking for investigators around the world who are experts in genetics and can collect samples for them, which we did. And so we built up this trust over years. Essentially, I worked for Glaxo when I was in Lausanne University Hospital for 3 years. And so they got to know me and get the trust that I deliver stuff and that I would be a good asset for them. If you don't have that, I think firstly, it's really important to explore, and coming back to my listening stuff, listen what industry needs. Don't try to feed industry by garbage. They don't care. They have their needs, and they will find the person who can meet their needs. So I would really encourage people, if they have an interest in going into industry, to first be very clear on what this company is looking for, and then tailor your application according to their needs and not yours. I think that's the very basic. Second, it takes time, as I said, and trying to meet industry people at conferences, for instance, is so important. So if you go to a conference on cancer, make sure that you have the chance to discuss maybe with the guys who are developing drugs for the very cancer you work on. And that takes time, builds up, listen. But yes, the first step in industry is difficult. I think it helps if somebody has already some track record in academia. It helps to build trust and confidence. I think that people shall not be in a hurry. They shall continuously be looking, and opportunities arise.
Well, I think that takes us to time. I'd just like to thank you for giving a great talk, and I'd like to pass over to Chiara to just wrap up today's meeting. Thank you.
Thank you.
Well, thank you, Professor Moser. Really inspiring, absolutely. And thanks for sharing your vision, your wisdom, your professional and personal life. Really, thanks a lot for taking time to be with us today. I really appreciate it. Thank you so much. And thanks also to Ravi and Paula. You have done an amazing job, really, in the moderation and in sharing also the junior investigator view on career. So thanks also to both of you. I just want to conclude this webinar with an announcement. As many of you know in the audience, we are also together with Rocio running another program supporting junior scientists in obtaining fellowship grant funded by the European Commission, the Marie Curie grants. Our next webinar will be on July 19, and we will give tips on how to develop a strong application for the Marie Curie Fellowship. If you need more information on the cost for Marie Curie program, you can check the EuroColangioNet website. You will find also the link to register for our next webinar. So I want to thank everyone again for being with us. I thank Rocio for co-hosting the webinar with me, and of course Vincenzo, University La Sapienza, for hosting us. And again, thanks Professor Moser for everything, really. Thank you.
Thanks a lot. Bye, everybody.
Thank you. Bye.
Thank you again. Bye.
Thank you, everyone.
Thank you.
Thank you.